It's now time to talk about the actual nuts and bolts of the meiotic process. Remember that the whole point of meiosis is to take what would be a normal diploid cell and have it go through cell division, but in the end up with cells that are haploid, cells that have half the number of chromosomes, or one member of every pair of chromosomes. The nice thing about this is that the stages are going to be the same names that you learned about in mitosis. So here, as we start meiosis, remember there are two cell divisions. So we're going to have meiosis 1 and then ultimately meiosis 2. The stages within each have the same names. Notice what we're starting with, prophase. Specifically because this is meiosis 1, this is going to be prophase 1. And really, as you look at the picture here, nothing should be new. The nucleus here is going to start to disintegrate. The chromosomes are going to become visible. The centrioles are moving towards the poles, and spindle fibers are forming between them. This is all stuff that you learned with mitosis during prophase. There is one thing within prophase 1 of meiosis 1 that is unique to meiosis. And to understand that, we need to look at this picture here. What you learned in the previous video was that we have 23 pairs of homologous chromosomes. What you're seeing here is one of those pairs. Here is a dyad chromosome on the left. Here's another dyad chromosome on the right, and they are homologous, which again means that they are the same but not identical. So if there's a gene, again, for eye color here, there's a gene for eye color here, but those genes might be for different eye colors. You can see based on the picture that something odd is happening. It is possible, now not going to happen all the time, but possible, that this pair of chromosomes can get tangled up with each other, and you can see that tangle occurring right here. What happens, or what can happen when they get tangled up, is that pieces of themselves can actually break off and swap spots. And so as you look at the picture down here below, you can see that the red chromosome on the left has a blue portion at the bottom, and the blue chromosome to the right has a red portion at the bottom because they swapped genetic information. Now, this doesn't seem off the top like it would be a good thing. How could that possibly be that the breaking and swapping of chromosomal material can be a good thing? Well, it actually is. Number one, let's address the fact that it's not harmful. If there is a gene here on this chromosome that deals with a certain trait, well, then there's that same gene on this chromosome that deals with the same trait. The genes themselves might not be identical, but if it's a gene that deals with the shape of your nose, then it's a gene that deals with the shape of your nose. It's the same on both chromosomes. So nothing was lost. They swapped homologous sets of DNA. Number two, it might actually, in the long run, be a helpful thing. If you think about a gene that might be, let's say, right there on that red chromosome, and another gene that might have been down here on what was the red chromosome, because those two genes are on the same chromosome, generation after generation after generation, they would always be passed on together. That is, unless crossing over occurs. Now, you have a gene that was part of this chromosome, and you have a gene that was part of the blue chromosome being passed on together. In other words, all I'm telling you is that crossing over leads to more genetic variety. And genetic variety in a species is healthy. The more genetic variety there is, the healthier the overall species is. So although crossing over is no guarantee, crossing over is, number one, not harmful. And number two, it can actually, in the long run, be beneficial. That takes us to metaphase, specifically metaphase 1 of meiosis 1. And although things are very similar to what you learned about in metaphase of mitosis, there is a significant difference here that you need to pay attention to. Now, you have the centrioles at the poles. That should sound familiar. You have the chromosomes lined up on the equator. That too should sound familiar. But look how they're lined up. They're not lined up single file. They are instead lined up in their homologous pairs. And so this blue one and this purple one must be one homologous pair, this orange and this red another. Notice also that each chromosome is only connected by one spindle fiber. This purple one on the right is connected to a spindle fiber on the right, but not anything to the left. And this blue one on the left is connected to a spindle fiber on the left, but it's not connected on the right. And therefore, what happens in anaphase is that the entire dyad chromosome gets pulled to the left here in the case of the blue chromosome, pulled to the right in the case of the purple one. The dyad chromosomes were not ripped in two. The homologous partners were separated from each other. The chromosome number is ultimately being divided in half, but the chromosomes aren't being split. So what you end up with here in the telophase cytokinesis combination of events is that you're going to end up with two cells. You can see the cleavage furrow taking place. The chromosome number has been split in half. In the picture here, we started with 4 chromosomes back there in prophase 1. And notice, once this cell gets divided in 2, you're going to end up with a cell over here that only has 2 chromosomes, and a cell over here that only has 2 chromosomes. So the chromosome number has been split in half. You've gone from a diploid cell to 2 haploid cells. And yet, we still have to have meiosis 2 occur. Why? Because these chromosomes are still dyad, not monad chromosomes. And that is the whole point of meiosis 2. Now, I'm not going to draw or talk about all those steps of meiosis 2. I want you to know that there's a prophase 2, metaphase 2, anaphase 2, and telophase 2/cytokinesis. But you don't need to learn those steps because you already have. They are exactly the same as the steps of mitosis. So prophase 2 is the same as prophase of mitosis. And metaphase 2 is the same as metaphase of mitosis, and so you already know them.